There are certain ways in which the body tries to prevent microorganisms, such as viruses and bacteria, from getting into the body. The skin is often the first line of defence. Not only does it act as a physical barrier, but the healthy bacteria found on the skin act as an extra layer of protection. The skin acting as a barrier also explains why it is important for platelets to clot breaks in the skin. Pathogens are most likely to enter the body in the air taken in by the lungs or by food and drink. This means there are certain defences to stop these pathogens. The nose is full of a sticky mucus substance which, along with the hairs in the nose, trap particles and stops them from entering the body. Likewise, in the trachea and bronchi, the mucus layer traps any particles that have entered the body with the air. The cilia will then work to remove the particles or pathogens from the body. If pathogens enter through food and drink, the stomach is able to destroy them. The stomach, as we already know, produces hydrochloric acid. This acid will destroy the pathogens present in the food. If the pathogens still make it past these defences, the body has another line of defence, known as the immune system. The immune system is made up of white blood cells which try and destroy the pathogens. There are different ways in which the white blood cells can destroy these pathogens. Phagocytosis is when the white blood cells ingest pathogens. They then destroy the pathogens. Some white blood cells are able to produce antibodies. Antibodies are produced specifically to destroy a certain type of bacteria or virus. Antibodies are pretty clever. Once they have been produced to destroy a certain type of pathogen once, they can be produced rapidly if the pathogen is encountered again. We are now going to take a look at how drugs are found. One of the places where drugs are extracted from is plants. Digitalis is a heart drug which is extracted from foxgloves. Willow trees are where the compounds for aspirin, a painkiller, originate from. Other drugs may be extracted from microorganisms. When Alexander Fleming was experimenting with culture plates, he discovered penicillin. One of the cultures with penicillium mould on had clear rings around the mould spots. This is why the drug is known as penicillin. New drugs are now being synthesised by chemists in the pharmaceutical industry. The starting points may still be compounds found from plants and organisms. A compound, for instance, may be adapted to suit the needs of the chemists. Before a new drug or treatment is used for treating diseases, they need to undergo a series of different tests. These tests make sure the drugs and treatments are safe for use. They also conduct tests to make sure the drugs are not toxic, to find the right amount to give people, and to make sure the drug gives the desired effects. Before clinical trials are carried out with new drugs, a set of preclinical trials must first be passed. Newly developed drugs are first tested in labs on cells and tissues, and then on live animals. Only when these tests are passed do the clinical trials take place. In clinical trials, human volunteers are used to find out the effects of using the drug. Low doses of the drugs are used at first to test for side effects. If the drug is deemed safe, then a select few patients with the disease are given the drug. This test ensures that the drug does the job it's supposed to do. A way of finding out the effectiveness of the drug is to carry out double-blind trials. In these trials, patients with the disease are asked to either take the new drug or a placebo. The placebos do not contain the new drug, but often a drug already in use to treat the disease. Both the doctors and patients will not know which drug has been given to which patient. This means the drugs can be compared without bias. The drugs can be used to treat patients once they have passed these tests. We are now going to look at how we treat diseases. Vaccinations are used to prevent illnesses occurring. A vaccination works off the principle that our white blood cells produce specific antibodies for each foreign antigen we come across. On the surface of each cell are proteins which are known as antigens. These antigens are different in our body cells to pathogens. When the white blood cells detect these, antibodies are produced to destroy the pathogens. A vaccine is made up of dead or inactive forms of the pathogen. When someone is given a vaccine, the white blood cells of the body produce antibodies to destroy the weakened pathogens. This means the white blood cells are able to reproduce the same antibodies quickly if the pathogens ever re-enter the body. This prevents further infection. Both bacterial and viral diseases, such as measles, can be vaccinated against. When a large number of the population is vaccinated against the same disease, the disease could disappear from the population. This is known as herd immunity. Antibiotics, such as penicillin, help to cure bacterial diseases. Antibiotics damage the bacterial cells which are in the body without harming the body cells. Specific bacteria are treated with certain antibiotics. Some antibiotics can treat a range of bacterial diseases, while some are more specific. Antibiotics are usually taken through a tablet, but they can also be used in injections. There are some issues with antibiotics. Firstly, they are unable to treat viral infections, only bacterial. This is because viruses reproduce inside body cells, and so it is difficult to develop drugs which destroy viruses and not the human cells. Secondly, the usefulness of antibiotics is decreasing. This is due to the number of antibiotic-resistant bacteria increasing. This increase in resistance could be due to the overuse of antibiotics. Scientists are trying to find new drugs due to this worrying resistance occurring. Other drugs, such as painkillers, relieve the symptoms of the disease, but they do not destroy